Buenos días. Todos, ya estás listo, ¿verdad? ¿Ahí atrás? Ok. Bueno, buenos días. Bienvenidos otra vez al segundo día. As we all know, in our very beloved school, there's always something happening. I was telling our guest about some of the issues that we have, especially the people right now that have these problems with their salary. We're just lucky and happy that they were so understanding to let us continue with the course. Hopefully tomorrow we will have, if maybe it's fixed and then there's not going to be any problem. If not, then we'll go through the same process that we did today, and we might start a little earlier tomorrow. I'll let you know, everything went up this morning at 5 a.m. is when I woke up, and then I got this announcement that the school's gonna be closed anyway. So anyway, thank you very much again. Thank you guys. It's yours.
Okay.
That's yours.
Hola. I just want to start by asking if there are any questions from yesterday before we transition to today's topic of transposons and their uses in genetics. Okay.
Can we ask the laboratory questions, or should we wait for or when you will review the paper, maybe?
I think
Yeah, okay, I'll wait
if it's a question that the whole audience could
relate to.
then I think it's fine. We're going to try to catch up a little bit because in the laboratory that's what we're using is transposons.
Okay, I'll wait then.
Okay.
Thank you.
All right. So, I'm going to give a little background as usual. I think I mentioned, I'm going to have a hard time remembering what I said in the lab versus what I said here. So, forgive me if I repeat myself too much. So, I think I mentioned historically E. coli used conjugation. Genetics. And salmonella transduction genetics. So, what do I mean by that? Transposons. Does anybody not know what transposons are? Yes? Okay. So, transposons are jumping genes. Okay? They are pieces of DNA that can move from one piece of DNA to another. Really, jumps just like genes. Okay. So there's three kinds. Insertion sequences. Composite transposons and non-composite. There are three things, three requirements to transpose. And sometimes instead of the word transpose, I might say a hop. Very good. Okay. So, let's start with insertion sequences. They're the simplest. A piece of DNA with a gene, an enzyme. So you need an enzyme. It's transposase. So ISs, IS, they're the simplest. They have ends for invert repeat segments, say 15 to 20 bases. And there is a gene inside that encodes the enzyme, the transposase. So the transposase gets made, and it recognizes these ends and allows it to then target, insert itself into the target DNA. Okay? So that's the simplest. And they're small, on the order of 1 kb. And then there's composite transposons, which are insertion sequences, the drug resistance, antibiotic resistance, usually, and another insertion sequence. So they have ends, and usually at least one of these has a functional transposase gene. Sometimes the others have some mutations, so it's usually only one of the two have a functional transposase. And so the transposase now can recognize the ends and transpose antibiotic resistance when it transposes. And then there's non-composite, which is simply an IS, but it has a transposase and an antibiotic resistance. So it's not composed of two insertion sequences that happen to land flanking something that it could then transpose. So those are the three classes. See? See?
Yes.
Now, this, I'm going to not talk anymore about non-composites. For genetic purposes, historically, the insertion sequences were important. Yeah.
How far do they have to be, at least, to be recognized as just one?
How far do they have to be? How far? Who are they?
The two sequences.
How far? Okay, that's a good question, right? So, for example, how far? Let's take the one I'm going to talk about, which is transposon number 10. We'll talk about that. And that has, let's call it IS10, insertion sequence number 10. And one of these, there's a one is on the right, and one is on the left. And this has T in B, A, and this has conditions. Okay, so the question is, how far does these have to be, or do just next to each other?
Next to each other.
Okay. It's really, the distance is how far the transposase can recognize. So in this case, this is about 10 kilobase pairs. So the transposase can recognize 10 kilobase pairs. Yeah. But if you look at transposition frequency and distance, 10 kb, 20 kb, etc., it goes down really fast. Okay, so it could be farther away, but the frequency is going to be super low. The exception that I talked about in the lab is an unusual transposon. And that is phage mu. Okay. So mu is a phage. Where's my phage? Not bad.
It's like a tree.
It's like a tree. It's got some DNA fruit inside. There's my chromosome. So mu inserts its DNA in the host, but it actually has a transposase gene which recognizes the ends and transposes into the chromosome. But mu is large. So it's a very large phage, but it has a DNA winding site. So there's an enzyme gyrase that winds DNA, and right in the middle of the mu is a perfect gyrase binding site. So that DNA gets in and it actually gets wound up. So it puts the ends right next to each other. So in that case, it can be as far as you want. Right? Pretty cool. Does that answer your question? All right, we'll talk a little bit more about mu later. Okay, let's see. Now, again, I want to go a little bit historically. ISs were important. So many of you may have heard of Barbara McClintock.
Yeah.
She won the Nobel Prize for transposons. She worked in corn, maize, and she saw the kernels varying, and her brain is much bigger than mine. Her papers are very difficult to read, but from following the color patterns in the corn kernels, she had this idea that the DNA was actually moving around into elements. So she came up with this concept of these movable DNA sequences back in the 30s. And very few people, I think, could read her papers. But the smart people could. And so she was worshipped by very few people. But then, later on in the 70s, there's a guy named Jim Shapiro. He was working on galactose operon in E. coli. And the gal operon, there's 3 genes. Okay? And it's not important what they are. And Jim Shapiro never had to write for grant money. Right? Isn't that great? Don't you wish you could do that?
Yeah.
Do you know why he never had to write for grant money? His grandfather invented the paper cup. He inherited a ton of them. Anyway, it's another story. So, Jim Shapiro. So I'm going to introduce you to a process that you may or may know about. So in bacteria, so DNA, RNA, protein. Okay. DNA polymerase. RNA polymerase. And this is ribosomes. Transcription. Translation. In bacteria, these are coupled. Right? Unlike eukaryotes, where in the nucleus you make RNA, and that RNA goes out to the endoplasmic reticulum, where it gets translated into protein. But in bacteria, when you make a +1, you make an RNA transcript, there's usually a region called a 5' untranslated region, and then an ATG, and then there's a stop, ATG, and then there's a stop, and an ATG, and then there's a 3' untranslated region. typical operant bacteria. And once RNA polymerase starts, the RNA polymerase could be in here somewhere, but the ribosome gets on right away. There's a ribosome binding site before each gene. Usually it's encoded at the end of the previous gene. Okay. So as soon as this transcript starts, you have your first, right, going down the DNA. You already have a ribosome transcript right behind it. Okay. So transcription and translation are covered. Now, there's something called transcriptional polarity. So, what is that? That happens. When you have a stop codon in a gene. Okay. So if we look at, this is kinase. If we look at kinase, and we look at mutations in the E gene, now E plus, kinase activity is 100%. If we have a missense, so something that prevents GALI from working, it doesn't fold properly, it's defective. But the ribosomes still make a defective protein. So the ribosomes are still right behind RNA polymerase all the way through. However, if there's a stop codon, so you might have 100%. But now we have a GALI amber stop codon. Sorry, let's stop. And I'll have 1, 2, stop 1, 2. 1 is early in the gene. This might have 10%. This is late in the gene. And this has like 80%. So this is what's known as transcriptional polarity. So when you have naked, so if the ribosome stops here, it doesn't come back on until the next ribosome binding site, right? So in the mRNA sequence, ribosome stops and stop. You have naked RNA. Okay. So normally, if the ribosome is all ribosomes are always covering the RNA, they're clothed. They're not naked. But naked RNA folds. So it folds in the secondary structures. You know, stem loops. Yeah. And that appears to be a mechanism that RNA polymerase has evolved to recognize. So sometimes RNA polymerase will just come on random DNA. Okay, so you have polymerase, and you have mRNA, but there's no ribosome binding site. It's not a normal start site. So sometimes it can make errors and just start anywhere. But if it's not translated, it's naked. And so this is going to fold, and it's going to make these secondary structures. And E. coli bacteria have evolved RNA polymerase to recognize these secondary structures behind and say, hey, this is not good. This is not right. It's not being translated. Off. Okay? And so that is an example of polarity. So when there's naked RNA, when there's a lot of naked RNA, there's a high probability that there's going to be an RNA fold that knocks off polymerase. But if you just have a little sequence, then the probability is much lower. Okay? What Jim found was a mutant in gal-E, which turned out to be an insertion sequence. So instead of having just a stop codon, he had an insertion sequence in E. So a large piece of DNA. And so nothing got through. When he had an insertion sequence, we had less than 1%. So absolute polar. It's because sooner or later, there was just stuff in there that would just knock off RNA polymerase all the time. So that was the discovery of insertion sequences in E. coli. And when they looked, they found that, in fact, there were big chunks of DNA that could move, that could hop, that could transpose from somewhere in the chromosome into the gal-E gene. So with the discovery of transposons, insertion sequences in E. coli, all of a sudden we realized that Barbara McClintock was way ahead of her time, and they gave her the Nobel Prize. All right.
Did Jim Shapiro got also the Nobel Prize or only Barbara Mike?
No, he didn't. No, he didn't. She'd already figured it out.
Yeah, yeah.
So once he saw this, he already knew from her literature what was going on. Right. He did help figure out the mechanism for transposition. There's a famous model called the Shapiro model to explain how it can move from one piece of DNA to another. We're not going to do that. Okay. All right, so a little history there. And that led to actually explain E. coli conjugation genes. So E. coli, here's my chromosome, infinitely big, has a plasmid called the F plasmid. And that F stands for fertility. And this F plasmid has two different origins. It has a replication origin called ori B for replication. It has another origin called ori T for transfer. It has genes for apellis, so it makes a structure that can attach to other bacteria. So when the cell divides, the replication origin fires along with the cell. So every daughter gets a chromosome and an F. They're coupled. But when it makes a pilus that contacts another cell, a cell that's F minus, that initiates replication of the transfer origin, and it'll transfer that F into a new recipient. So this is conjugation. So far, so good. No questions yet? You know all this? No. Some? They're supposed to. Okay. So how does this get to conjugation genetics? So the F plasmid has insertion sequences. IS2, another one called IS3. Insertion sequences in the F plasmid. And the E. coli chromosome, I think, I'm not going to get the numbers right, but IS2, IS3, there's about 30-ish. So there's a lot of insertion sequences throughout the E. coli chromosome. And what people have found earlier was that this F plasmid, using recombination, could recognize by recombination the REC-A protein, could recombine between IS insertions in the F and IS insertions in the chromosome. And that generates the F. Inside the chromosome of E. coli. And this is called an HFR, or high frequency of DNA transfer. Now, this still makes a villus. It still has an ORET. It replicates when the chromosome replicates because it's in the chromosome. But if it recognizes another cell, it can now transfer. It doesn't know it's in the chromosome. So it just starts transferring DNA. And it'll keep going. So this could be the DIS, this could be TRIP, this could be pure E. So it starts bringing in DNA that has a piece of the F, but then chromosomal DNA. And that can be used to recombine. Let's say this is S minus, plus. It can be used to transfer DNA from a donor into a recipient. So that's how conjugation genetics came about in E. coli, and it was dependent on these insertion sequences, being in the chromosome and in the F. Yes?
How? What about the element integrated and conjugate, like a transposon or R-class?
On our class, I'm going to talk about our class. This is basically an R plasmid without a resistance gene.
That is not a nice integrated conjugate element.
R?
I think conjugated transposons,
Yes.
maybe, so she's talking about.
Okay. So I don't really know much about conjugated transposons. You know?
Fundamentally, it's a transposon that carries conjugate elements inside of the transposon.
I see. So a conjugated transposon will be a transposon that can insert and has an ORET,
Yeah.
and that allows it to transfer just using the information of transposon. Okay. Thank you. Good. All right. So where are we going with this? I don't want to say too much, but well, I will say something that if so this takes, to go around the whole chromosome, it takes 100 minutes. So it takes 100 minutes to transfer the entire E. coli chromosome, and historically, that's why the chromosome is 0 to 100. It's 100 minutes because of the time of transfer. And just for historical reasons, right at the very beginning is always the threonine operon, whereas most other chromosomes, they have the origin of the replication, whereas in E. coli is somewhere over here. That's just a historical artifact. Okay. Okay, let's get back. One last thing. If you have, let's see, let's draw this. One last thing. Let's have our F with our origin of transfer here. So you can insert this anywhere around the chromosome. That can transfer different genes. But we can have a situation transferring to this place. So here's a strain that could go all the way around the chromosome before it gets to histidine. But since there are insertion sequences all over the chromosome, you can put these in a blender and this could take maybe 100 minutes to get all the way around the histidine. But if you put them in the blender and you do it after 10 minutes, this breaks. So if you select for the transfer to his plus, what can happen is these can come out. So this is reversible. But it can come out here as well. It comes out here, you get and these are called F primes. So they can carry parts of the chromosome. And that allows the E. coli geneticist to do complementation. So that's how Jacob and Minot were able to make 2 copies of the lac operon by having one on F and one on the chromosome, and they could do complementation. Okay, very good. So I think that's all I want to say about E. coli. All right. All right, let's get back to transposons. But and they didn't know they were working with transposons. They just called those affinity sites for integration of the F. All right. So let's get to genetics. All right.
So that means that the whole chromosome is transferred to the new one.
You can transfer the entire.
How stable is that cells with the double?
Oh, okay, that's a good question. So let's bring in. Yeah, you can bring in the whole chromosome, and it's going to get degraded, and it's also going to get pieces are going to get recombined in. Okay. So you're going to have a whole lot of recombination. Yeah.
The question is, if you bring in the whole thing in a little bit, it could circularize by recombination. And now E. coli is a diploid, a genuine diploid.
That
Is that stable? That's the question.
Yeah.
The answer is no, as far as I know. Yeah. You would then divide and only just like regular division. I mean, E. coli, when you grow E. coli in LB, it's got more than one genome. If you grow it fast, you can get many genomes before it divides.
But I don't think that's possible what Benjamin is saying, because as soon as the DNA is starting to come into the cell, it starts to recombine. So there's not going to be any chance that you have a whole molecule of DNA together and bind to form a
However,
carbon.
but this is reciprocal. So if
Yeah,
this
yeah.
comes in, this goes up here. So
Okay.
it's conceivably possible.
Conceivable? Okay.
Yeah.
You can do it, it's just rare.
Yeah.
And they're not stable. So,
Okay. Anything else that I don't know about? All right. Okay. Now let's get back to transposons. So for genetics, most popular transposon is TN10, TN5, U. These are the main transposons that people use. These were discovered originally in our plasmids. Right? So resistance plasmids. In this case, it's tetracycline resistance. This is kanamycin resistance. And this is just a phase. We'll talk about that. Okay, so for genetic purposes, this is really great. Because if you can introduce the TN10, and this was originally done in E. coli, on phage lambda, where there'd be a lambda with a TN10. And that lambda had mutations in replication genes, not codons, such that they could only replicate in certain cells. If you put them in a cell where they can't replicate, then the phage dies. But the transposon can hop in the chromosome, and you get tetracycline resistant tolerance. And when it lands in a gene, so TN10 is 10 kilobase pairs. If the TN10 lands inside the gene X, what happens, and this is now X base. So you have a huge disruption inside the gene. All right. All right. So we can do this and disrupt any gene, any non-essential gene. So if we do look on a plate, L plus N, and we plate the colonies. Each colony has a TM10 at different positions in the chromosome. So it transposes, not completely random, but random enough. So each colony represents a different insertion around the chromosome.
When the transposase inserts the transposome into the genome,
Yeah.
it recognizes like specific sequences, or it's
So TN10 has, so if we look at a gene, let's say LAXI, and you look for TN10 insertions in LAXI, you will find hotspots. So there are target sequences that seem to be preferred, but not so bad. We can't work with it. There are mutants that Nancy Pfeffer has isolated in the TMPA gene, the transposase gene, that are called ATS. They're altered target specificity. And with these mutants, now this one. So you can make mutants in the transposase that lose that targeting specificity. They become more random. Alright. So this frequency of transposition is not very high. It's pretty low for TN10. It's only 1 out of 10 to the 5th. So every, 100,000 times this gets in, only 1 is going to transpose. But if you work with bacteria, we can add 10 to the 8th phage plus 10 to the 8th cells, and we get 1,000. Yes? So even if this frequency is relatively low, we work with huge numbers. That's why we work with bacteria. We can work with large numbers in itty bitty space. But the advantage here, though, is once you have it in, it's pretty stable. So let's take, for example, this insertion. We made this insertion in the X gene. Now, if we move that into a new recipient, we move that DNA in, it's only going to transpose at 1 in 10 to the 5th. So if we select for tetracycline resistance, we are going to get it primarily by recombination. So the recombination system of the cell is going to use flanking homologous DNA to bring this into the chromosome. And we get back in. So transposons were fantastic because now we have a marker gene that we're interested in, X, and we want X minus, we can insert this transposon in it, and that transposon, that X minus, is always marked with a drug resistance. So we can then move it from strain to strain, bring in tetracycline resistance, and we know that we knocked out the X. So that was really powerful. when Stanley was talking about point mutants, right, that is just a missense, how are you going to get it from one strain to another if there's no way to select for it? So transposons provided selection. And as Stanley said yesterday, selection is worth 1,000 screens. That's the power. So genetic transposons really transform bacterial genetics. All right.
But even though there are several hotspots, each one has a different frequency.
Each hotspot.
So you have several points along the chromosome that can recombine, but not all of them has the same frequency, right?
I'm not sure I understand your question. Yeah.
So I think this gets to a brief mention of TN7. It has very high site specificity, and TN10 has pretty low site specificity.
Right. Stanley's talking about a transposon called TN7. It's a transposon that'll hop into one site only. That's a super hotspot. But I don't think that's your question.
No, with TN10, in every 1,000 bases, you're likely to find its preferred site.
Oh, right.
Right? So that's the key.
Within any gene.
Sorry.
There'll be with TN10 some hotspots. Is that?
Yeah.
Within any gene. Unless you're working with the one that I mentioned, which is the ATS, the mutant in the transposase, that loses that specificity. Then it becomes more random. So with any gene, it'll be all over the place.
So all the sequence has the same frequency.
In this case, yeah.
With that mutant.
With that, with the wild type, you'll have hotspots at maybe 10-fold higher frequency than anywhere else in the gene.
Oh,
Great.
okay.
Anybody else? So, let's see. All right. So it's really great because when we do the hop, when you do that hop in an LTEC, again, each colony represents an insertion in a different place in the chromosome. Yes?
Only with the mutant.
What's that?
With the mutant. With the wild type.
You know, with the wild type, it's not that bad. I mean, it's really not that bad. I mean, if it's in the same place, the same site, if it's in the same site, it was an independent event, right? So it went in, and maybe, yeah, there's a higher hotspot where it'll be more frequent. But it's still a different transposition event. It's still a different bacteria that got the transposon. But for the most part, the whole chromosome gets hit.
Because if you would like to use this transmission system for your screening of any gene,
Yeah.
all the sequence, you would like to have this same frequency.
It depends on what you want. If you just want, what do you want? I mean, what do we want? If we want vaccine or lactine tablet, we want to knock out the lactose gene, lactose utilization. Right? We introduce TN10, allow it to hop, and it'll hop all over the chromosome. Right? And there's 5,000 genes. The odds of landing in any gene is going to be any gene. 1 in 5,000. So you have to look at the probability of getting into any gene. It's going to be 1 in 5,000. Yeah.
ask you a question, Kelly,
Yeah.
that ties to this? All of your searches for TN10 insertions near your are in your favorite gene. Have you ever found them in the same site?
No. That's
And
true.
how many have you looked at?
But I mean, I haven't done what I mentioned. People that are like looking, they look for hotspots. I mean, they're looking for that.
Right.
I just want to get an insertion in my gene that I can work with. Once I have one, I don't need 2 or 3 or 4, right? I just need one. So if I'm looking at, I work on a flagellar system. So if I have a gene for flagella, I don't need it throughout the gene. I just need one. So if I can then move that mutation into a new background, I always bring in the minus for that gene. I only need one for what most people do. Because I'm not studying transposition into my gene. That's somebody else who's done all those studies. I don't need to do that.
Yeah, you just practical. The use of the transposons is practical.
Exactly.
And then you look for a phenotype to be interrupted or to appear, depending what you want.
Right.
That's when you use the transposon.
Right.
So maybe it's worth it sometimes to know the frequency, but if somebody else figure it out, then you just use the tools like that ATS mutant for the transposons.
Sure.
That is very helpful.
Yeah.
So then you don't really need to care how frequent that happens.
Correct.
Isn't it?
Yeah. That's correct. Okay. So, how do we find these? If we want lac, again, if there's 5,000 genes, the odds of getting into that gene is 1 in 5,000. But the rule of thumb is if you want a greater than 99% probability, then you have to look through 50,000. Which, not so bad. I've done it. If you have a positive selection, let's say I go onto place, and it's 6-amino nicotinamide, and I do a TN10 hop, I simply look for guys that grow. So if I do it on L, I can do it so that I have 10,000 per plate times 5 plates, and I replica print and look for colonies that grow. Now I've got, say, a pink A, N, E, N. So if I have a positive selection, that makes it very easy, right? Of course. But what if I don't have a positive selection? If I want a proline TN10, let's lowercase, proline TN10. I'm looking at proline utilization, proline biosynthesis. Then I have to do, maybe 500 per plate. And then I have to replica print. Okay. And it's doable, it's not hard, right? I've done things where I've had to do 500 plates. You do it in one day. And we go on to a minimal glucose. And then a minimal glucose plus proline. And you look for the guy that only grows. Here. So here's the mutant that only grows on the plate with proline. And you clean it up, purify, and that is going to be and now, once we move that into a recipient, and we select cycling, Again, the frequency of transposition, if it's 10 to the minus 5, these guys are going to be essentially 100% pro minus. Right? So then the drug and the phenotype is 100% linked. Right? Okay. What about something that's a little harder, like just the point one that Stanley talked about. So we have one that we've worked with in a serine tRNA. That's TS. Dead at 42 degrees. So it's a missense mutation that causes the tRNA to unfold at 42, and it's essential tRNA, they die if it can't read these serine proteins. What if I want to move that into a new string? Okay, so what I can do is I can take my serine TS and I can bring in TN10. And I can do my, I can plate them on LTET. What temperature am I going to incubate these plates at? 30 degrees. Okay. And again, these guys are going to go all over the chromosome, but sometimes they're going to go near my gene. Okay. Okay, so I can take these guys and I can pool these cells. Now, if I move, and we haven't talked about transduction, class guys, but if I move, talk about transduction. So random TN10s can be anywhere in the chromosome. So sometimes this will be over here, nothing, I'm still CRT plus. But if I have a TN10 that happens to be near my CRT TS, I can bring in tetracycline resistance and my marker. Okay. So I can look for guys at 30 degrees. And then look at 42 degrees. I replicate for things. And now I have the TN10 that's near it, so that it can bring in the tetracycline that's next to the TS. So now I can move around markers, right? There's an easier way. An easier way is just to take wild type cells and do the, got that right. Again, do our TN10 log. And then I can pool, scrape these all together into a pool, and my pools are typically 200,000 colonies. So with this number, I pretty much have an insertion in every gene in the chromosome, right? That's a good pool. All right. And then from that pool, I can go into my CRT-TS because this is a wild type. I can't insert T because it's an essential gene, but I can insert next to it. Now, I bring in plus. That's easy, right? Now I can do my L tet at 30. And then I can replica print, move it on to 42. Right? So now it's easier because I'm bringing in the one that's going to grow as opposed to the one that's not going to grow. So this is more of a selection than a screen, right? But I am screening for CRT+. And with that, so now I have a TN10 that's near CRT+. And I can use that to go back in to my CRT-TS. And I select for TET, but I screen to keep this. So TET resistance, but 42 degree minus. Okay, and depending on how close this is, will depend on the frequency. So if it's very close, then it's going to be a low frequency that I keep this. But if it's further away, it's going to be easier. 10, 20, 30, 50%. Depending on how far away the marker is. Okay, time.
Great time.
Break time. All right. Break time. Any questions? Right. Right. Shall we take a break?
Yeah.
Okay.
20 minutes? Yes. 20?
Fine.
So 20 minutes, I think the coffee is ready outside, so you guys wanna
Kelly forgot to say, and he asked me to say, to remind that the other cell, the cells come together until they retract. I think you all know that, but they come together, and that's how the DNA transfers. All right. Kelly said a number of things I'd like to just comment on. One of the things he talked about was Barbara McClintock. Both of us have been in a very interesting place in the world because from the time we were graduate students, we've been able to meet some people who were really famous from the previous generation. And one time, I was at Cold Spring Harbor Laboratory, and they have a dining room. And I was sitting at a table eating, and this short little woman comes up and sits and starts talking to me. And we had a wonderful time talking. And then she invited me back to her house for tea. And every time I was there at Cold Spring Harbor, she invited me to come with her and talk in her house, and we had tea together. And that was Barbara McClintock, and she had a Nobel Prize, and I didn't know who she was. But we developed a lifelong friendship. So it says something about science, and how science can be such a small place. Another person I got to know really well was a French woman named Lucia Hirschby. And Lucia is probably not a name that you know. But somebody asked the question about diploids, right? And Lucia studied a situation in Bacillus subtilis where you get something that she called non-complementing diploids. And the idea was that sometimes they could find cells that seemed to be diploid, but they didn't complement. It didn't make sense. She made a complicated theory that these non-complementing diploids, where one chromosome got turned off and the other stayed on, like what happens with the X chromosome in humans. Yeah? It ends up, this was some years ago, it ends up that that work was entirely wrong, not because they did anything wrong, but what happens is some of the bacillus get a property and they get really, really sticky. And so you have two cells sticking together, and when you plate them, they form one colony, right? And so in fact, these were two cells that were stuck together, and that's why they couldn't complement. But they couldn't see that. At the time, they didn't have the optics to be able to see the individual cells like that, right? And I think that's one of the lessons of no matter how hard you study a subject, there's always something else you should know. I got to know her really well too. She lived in Paris. She lived on the 7th floor of an old building that looked down on the quad in Paris with the guillotine. So, interesting experiences. I'm going to talk to you, I'm going to follow up on what Kelly talked about and talk to you about portable regions of homology. But before we do that, I think we should talk about homology on the chromosome in general. Okay? So we think of bacterial chromosomes as being these structures. We draw a circular chromosome where genes are in certain places. But chromosomes change a lot. They are not stable structures, right? So if you think this is just a small part of a chromosome I'm drawing, where these are different genes, okay? And the concept here is that there's a small region in the chromosome that has homology. These are chromosomal genes. When this happens in the chromosome, a number of things can happen, right? So one of the things that can happen is as the chromosome replicates, here's a replicating chromosome. As it's doing this, what we end up with in the cell is something that looks like this. Just change color so it's easier to follow. So now what we have is, out here, here's A. This is the first BC. Now, there's D, E, D, E, B, C, B, C, F, G, F, G. This is a replicating fork, okay? We've replicated, and in the cell, at the moment of replication, these regions are diploid. Yeah? Now when these regions are more than about 10 base pairs, they can recombine. And that can do two things. In one case, you can get a recombination event that looks like this, a recombination between here and here. And if that happens, let's just draw it out. Here we come. We get A, B, C, D, E, B, C, D, E, B, C, F, G. We have just duplicated a part of the chromosome. Right? This is chromosomal genes. No transposons have to be involved. Right? This is simply because recombination can happen, and it does happen. Now, this is a duplication, but once we've duplicated, it can duplicate. And that can duplicate. And so in cells, under some conditions, you can accumulate many copies of a region of the chromosome. Yeah? When that happens, and you get many, many copies, it's called gene amplification. Okay? Remember the other day when I was talking about antibiotic resistance? One of the things I said can happen is that you can get gene amplification. Right? So let's just say that this D gene gives you very tiny amounts of antibiotic resistance. Right? Very tiny amounts. But now, if we amplify it, so in the cell there are 50 copies, now we have full-blown antibiotic resistance. We have 50 times the tiny amount. Right? So there are certain things in the cell that will select for these duplications and amplifications. Right? If you ask cell to grow on a carbon source at very low amounts, sometimes the genes that use that carbon source, if you amplify them, they'll grow better than the others. So that can happen normally.
I just wanted to say that that's also an example of how you can evolve a new gene. If your D has some other activity at 1%, and then you select for that activity, and you get 100 copies, now that one could mutate to maybe go to 5%, and then higher, and now you have until you have a new gene.
Makes sense? Because the fact that it's in 2 copies means each copy can evolve separately, right? So, anyway, chromosomes do this a lot. When you have a growth condition that they can grow faster, if they amplify some region of the chromosome, that region will get amplified. Okay? When you put them in rich growth media, very often these will collapse and go the opposite way. How do you go the opposite way? Well, it's not so hard. Look at it. Now, let's just take here, and now we get a recombination event between here and here. Now we come down, we get A, B, C, F, G. We got a deletion. You can duplicate, you can delete. Chromosomes are doing this all the time. Yeah? Okay. Now, I have to say, as a rule, a lot of the things we do in bacteriogenetics are based on the idea that they're a haploid, right? So, under those conditions, we're looking at the haploids. But if we force cells to do certain things, they will, they can, and they do amplify. Okay? The one region that you can't duplicate like this is the terminus of replication, right? Because if you duplicate it and there were two termini, you come around, the DNA stops here and it can never get to the end. It'll stop at both places. So if you try to duplicate the terminus of replication, the cells die. Okay. Oh, by the way, these things can be extremely large, very large. You can duplicate a third of the chromosome, a half the chromosome. You obviously can't delete half the chromosome. You can't delete any part where there's an essential gene. And in every chromosome, if you divide it into 100 minutes, there's an essential gene in almost every 1 minute of that chromosome. So there are limits to deletions. All right. Those are recombination events that happen on the chromosome and happen normally. But what if we want to engineer chromosomes, right? So we make changes that we care about. And that's where transposons can be really important for us to use as portable regions of homology where we can put whatever we want. Okay? Everybody on board? Everything okay so far? All right. So TN10 is really a very nice transposon for this, but mud is also a nice transposon because unlike TN7, we can find these insertions all around the chromosome. Right? So I'm going to use TN10 as an example. In a little bit, Kelly's going to give you an example using mud. Right?
Really?
In one of the papers.
Ross doesn't know about mud, just the lab.
Oh, well, you're going to have to make it clearer than mud. So, I was going to tell you about insertions near genes, but Kelly already told you about insertions near genes. So let's just say, he mentioned to you that HFRs can be really valuable for moving segments of the chromosome, right? So wouldn't it be nice if this is the generic chromosome of Salmonella. Let's say what we would like to do is we would like to make HSFR insertions at different places. Maybe we would like one here, and we would like one here. And they don't exist. So how can we do that? Yeah? So one way of doing that is we have an F prime. Okay, so it has an origin of transfer. F prime. And this F prime has a temperature-sensitive origin of replication. And it has a TN10. Okay? This is constructed. Now, let's say we want to have an insertion right here. We use a strain that has a TN10 at that location on the chromosome. Or, sorry, no. This is lac plus. The Salmonella chromosome is lac minus. Now, all we have to do is we have to say, let's bring this F prime into this cell, and we're going to force these cells to grow on lactose at 42 degrees. Okay. At 42 degrees, this cannot exist as an independent plasmid because it can't replicate. Right? There's no lac plus here, so you can't simply lose that plasmid. So what happens is you get a recombination event in the two TN10s, and that integrates this into the chromosome. Let's just draw that out. Right? So let's just start right here on the chromosome. We're going to come up, we're going to get part of TN10, we're going to get the rest of TN10, we're going to come around, down, and out. So our new F prime, at the specific site that we had that TN10 to begin with, right? So we're going to, I'm going to put A, B, C, D, E, F. So we're going to have A, B, C, right? We hit the first part of the TN10, we come up, we get the F prime, pointed in that direction, we get the next part of the TN10, come down, and we get DEF. So easy. So easy, and we have put that origin of transfer exactly where we wanted it on the chromosome. Right? Pretty cool, huh? This, we couldn't do things like this. They were really hard to do before we had transposons, as Kelly mentioned earlier. So the people who figured out how transposons work opened up the doors for doing a lot of sophisticated genetics. So this is a way we can put an F prime wherever we want. If this TN10 was anywhere in the chromosome, the same thing can work. Okay? All right. I have to erase a lot more because I write a lot bigger than Kelly. So, all right, so that's a portable region of homology. We put the TN10 where we wanted that HFR, not where it wanted to go, where we wanted it to go, right? So now let's, did you talk about localized mutagenesis? Right. So now let's say we have a situation. Here's a cell. I'll take one of my favorite regions of the cell. This is where the proline, oh no, I forget. Proline utilization operon. Proline utilization operon has a nasty name in Spanish, so we won't do it.
Oh, right.
Oh,
Just use it. It's fine. Just use it.
Okay. Yeah, I'll put in the transport. All right. So this is a set of genes that maps out here on the chromosome. So I'm always making the chromosome where at the top is 0 and 1, right? So I was really interested. This is a very interesting regulatory system that involved molecules shuttling between cytoplasm and the membrane. And I wanted to understand it, so I wanted a lot of mutations in these genes, right? So to find a lot of mutations, I used something called localized mutagenesis. Right? So I started with a strain that has a TN10 that's near the PUT-PA operon, but not in it. Near it, not in it. Okay? And what I did with this guy was I took it and I grew phage P22, which Kelly has talked to you about. So now, in some of those cases, packaged inside of P22 particles, I will have this TN10 and PUT-PA+. Okay? In other of these phage particles, I'll have just different regions of the chromosome. from all over the chromosome. But some of them will carry this region right here. Now, I can take this whole group of phage, and I can now mutagenize it with a mutagen like hydroxylamine. And that causes point mutations in DNA. Okay? So within these phage, all the phage are getting mutagenized, right? But I only care about these ones. Some of the phage will pick up a point mutation in put P. Some of them may pick up a point mutation in put A. Some might pick up one in the region next to it. But I mutagenize at a level that each phage only gets one hit, most likely. Okay. Now I take this phage and I transduce them into my put PA plus cell, and I select for the TN10, so I demand tetracycline resistance, and sometimes it will bring in the put PA minus mutants. So I select for tetracycline resistance, and then I screen for put minus mutants. Right? And because I know that I have a high frequency of mutagenesis here, I have a high probability of finding my unusual kind of mutant. And I haven't mutated anywhere else on the chromosome. Right? So this is called localized mutagenesis. There are some slightly different ways you can do it, but you can see I'm mutagenizing a very small part of the chromosome. So in the lab, Kelly told everyone that when P22 packages DNA, it packages about 1% of the chromosome. So this is a pretty small region. I wanted to find mutations that change the ability of a transport protein to recognize sodium, to change it so it recognizes lithium instead. That's a tiny, tiny change. It's really hard to find. Without doing this high level of mutagenesis, I never would have found it. But we did. Well, actually, I didn't find it, my student found it. All right. Yeah.
When you change a base for another by hydroxylamine,
Yeah.
how you can control that only one pair of bases?
So when you do mutagenesis with any mutagen, you can actually measure a killing curve of a region. And if you back up on that, you can figure out the frequency of mutations versus the probability of killing. So you can base it on that.
It's exposure time.
Yeah, absolutely. So it's the amount of time you're exposed to the mutagen and the concentration of the mutagen that determine it. But you determine that ahead. Once you've done it with this kind of phage, you could use that same thing for any region of the chromosome. Right? So if Kelly wanted to come in and mutagenize his down here, all he would need is a strain that has a TN10 near his to be able to do that same thing.
Is there a library of DNA mutants in different non-essential genes?
There is. Yeah. There's a library of mutants.
Where can we get it?
Well, the easiest way to get that library of mutants is to ask Kelly.
We might you up here tonight.
No, people who do serious salmonella genetics will have whole freezers full of these, right? I once had a good freezer full of it, but my freezer died and I lost a lot of strains, so he has better collection than I do. Were you going to ask something else about the question?
No, no. That's it.
There's a,
Thank you.
I could give us another 5-minute answer to a question, but maybe that's not what you asked, so I won't. Was there another question? Right. Okay, yes.
But that mutagenesis is random, right? Inside the entire gene?
Inside of just these phage particles, that can happen anywhere.
And can we control which base pair is mutated?
No.
No. You have to.
So this is why we're selecting for TN10 and we're screening for our desired mutants.
But you can control it so that the probability of that particle having only, you know what exposure gives you only one base pair change per particle.
But we don't know which base
You won't know, then you have to screen.
Okay.
Yeah, screen.
Actually, let me back up for a second. Let's say I didn't know anything about these genes. In that case, I might want to start and heavily mutagenize that region. So I could look for a whole bunch of different mutants. But I'm looking for really special kinds of mutants. Then I want something that's not mutagenized as much so that I don't have to worry about multiple mutation effects. Let me just say one other thing. This is pretty sophisticated genetics, but this is pretty easy to do. Yeah? I mean, all you need is some phage, the transposon, and a way of selecting for the transposon, in this case TET resistance. So you have to have TET plates, and a way of screening for your gene of interest. Okay? Easy. That's what's so, this is why genetics is so much fun. You do a lot of thinking about what you want and what you think it's going to tell you, but the way you go about it, that's not so hard. Okay. Now let's say, all right, I have found my mutant. And my mutant, at my particular mutation, is constitutive for this PUT-A repressor. Okay? Sorry, for the A gene that encodes the repressor. Constitutive. Okay? And I want to be able to understand, is that a dominant or recessive mutation? Right? So to be able to do that, I would really like to have that region diploid, right? So how can I use a TN10 to make a diploid region? So here's our bacterium, the recipient. And I'm going to just call this A plus, B plus, and then I'm going to put a TN10 right here, and A plus, right here. Okay? Now, I'm going to start, I also have other strains that have TN10 on this side. Okay? So I'm going to grow a phage on one of the other strains, and it looks like this. Right? So this is the bacteriophage. This is the recipient chromosome. There's a TN10 encoding tetracycline resistance on both. And this is the thing. Okay? Now, in this case, I want to look at a mutant that's constitutive, right? So I'm going to start with this constitutive allele. Those two guys are a little different. Now what I'm going to do is, I know how hard it is when you're taking notes and somebody erases things. selectable marker in there. That's right. I was a little sloppy or something. Okay, so now what we can have happen is we're going to demand that the cells are purity plus. You have to be able to grow without any added uracil. Okay? This is pyrimidine biosynthesis. And that they're tetracycline resistance. One way we can get that is to get a mutation here, and a mutation here. And so now the product, we come down, we get part. No. Everybody go to sleep for 2 minutes. Close your eyes. Let me try more. Hey, there you go. Kelly, you teapot.
I have to talk about it first.
Yeah, that's true.
Talk about it later.
Yeah, go ahead and save me the embarrassment. Okay, let's see where
¿Tienen el aire o estamos bien así?
No, sí estamos bien.
¿Sí? ¿Quieren abrir la puerta?
Are you ready?
Enough distraction?
No, I got it.
I mean, we got this distraction enough?
Yeah, that was good. Okay, so if we get a recombination event here to here, look at what we have. Here's our pade constitution mutation. We come down. This is pyrimidine D minus. We come back. Here's the tet. Here's the pade plus gene, and this provides pyrimidine D plus. Okay? So now we have a recombination event on this side. So we duplicated that specific region that's flanked by the TN10. So our new strain, we draw it out. Here we go. But P plus, A, constitutive, pure D minus. Here's our TN10. Here, comes up here. Here we have but P plus, but A plus, pure D plus. Okay? Now we can ask about dominance. A plus over A constitutive. Very simple to do, as long as you draw your things right. What I had done originally was I put my transposons on the wrong side, so I made a deletion of this entire region. You see that? A duplication and a deletion are just the opposite kinds of things, right? It just depends on what your donor has versus what your recipient has. Make sense?
Yes.
So look, easy to do complementation analysis in that small region of the chromosome without building any special plasmids. Let me tell you one other thing that's really important here. Look, I have one copy of A plus, one copy of constitutive. 1 to 1. So if I'm asking about complementation analysis, they are both equal. If I brought in a multi-copy plasmid that had 20 copies of A plus and 1 copy of constitutive, maybe the result I'm getting simply because of heterodimerization. Right? So this is the cleanest possible way to do complementation analysis. Right?
And it's reversible. So if Stanley takes away selection, so tetracycline resistance, if he grows them without selection, it'll come back out. And it can come back out leading haploids. One haploid is AC, one haploid is A plus. That way you know you did the complementation. Okay? If the two mutant types come back out, then you know that the 2 mutant types were in the duplication.
And a really nice thing about tetracycline that's not true of a lot of other cases is we have a way of selecting for tetracycline resistance, growth on TET, but we can also select for tetracycline sensitivity, requiring growth on plates that have fuchseric acid.
Is this, I guess this is the protocol to follow with that work that you gave us. The paper that you published with Guido about moving different regions between salmonella typhi muon and typhi.
Yeah.
So yeah, so there's also a conference that is on YouTube on using these technologies. So to compare between typhi and typhi muon, just to see which genes were important for each one of the serotypes. So I'm gonna send it to you in case you wanna see it. So I don't remember when was that, maybe a couple of years ago? No, it wasn't COVID time.
No,
Maybe
before COVID.
Before?
I think it was before COVID.
I'll look for it and then send it to you.
Everything important happened before COVID. And I'm going to quit here just a second. So yeah, there are a number of other tricks that you can do using these transposons, just by putting them in the right place on the chromosome. So the kind of transposon libraries that Kelly described, where you have the 200,000 insertions, so you can find one virtually anywhere in the chromosome, that is golden. If you have that, you can do a lot of different things. Okay? And just to follow up on the last comment, Salmonella typhimurium is the tool horse for all of this genetics. Salmonella typhi, now, at a certain point, they didn't have any genetics. It was very hard to study. But it's really important because typhi causes serious human disease that's different than the disease caused by typhimurium. Right? And so by knowing these genetic tricks, we figured out ways to move those tricks into typhi. And if any of you are working on some other bacterium where you don't right now have genetics, the possibility exists if you think hard and you involve somebody who's done a lot of this genetics to develop those techniques for your organism. Right? And I'm going to stop right here. I didn't tell you about levitation. Does levitation translate?
Levitation.
Yeah. In Spanish, levitation, which, of course, everybody wants to know how to do, but I'm not going to tell you about it. You're on, Philip.
Okay. Oops. I have my computer. Alright. I have to tell the class briefly about mud and then teapots, which the lab already knows about, so it'll be a little bit, I'm going to shorten down a little bit. I mentioned phage mu. Phage mu is a transposon, and when that phage enters the cell, it transposes into the chromosome. So mu stands for mutate. And it can go anywhere. That's a gene X. Then we have an X with a mu insertion. So this is going to be mu minus, right? And that's becomes what we call a lysosome. So it can stay in the chromosome, or it can replicate. And it replicates by transposing around the chromosome to make maybe 50 copies. And then those get packaged into new heads. And they come out. So you can get a simple insertion or a lytic growth. Those are two ways of transfer. So mu, draw it like that, 37 kilobase pairs. And it has transposase, the A gene, and then at one end there's tail genes. And what a guy named Malcolm Casadevine did, and so this mu can transpose like to anywhere in the chromosome. Of course, if it transposes into a central gene, that it senses that the cell is going to die, and then it does the rest, so it gets out. Okay. So a guy named Malcolm Casadevine cut this part out, and he replaced it. I'm going to talk about just one with the lac operon. It's true, I don't write straight. And an ampicillin resistance gene. So that provides a selection. So this lac operon has no promoter. And he used this as a way of putting promoters of genes regulating lac. So now, if this inserts into that gene X, and he can select for those insertions because they confer ampicillin resistance to the cell. And now the lac operon has no promoter. In this case, it has a ribosome binding site for X, but the transcription of the lac operon is dependent on that promoter. So positive or negative regulators of that gene X are now either positively or negatively regulating the lac operon. Okay. So that's great. And should I talk about duplication? Mud-generated duplication. Okay. So you can actually use this like Stanley mentioned with TN10 to generate duplications over any region of the chromosome. That you can read the paper, maybe we can do that. Read the material. Okay. All right. And there are versions of this. That's called MUD1. And as a grad student, I actually made an AMBER mutation in the A transposase gene. So only in a strain that had a tRNA suppressor gene, RNA, that could read a stop codon, could this transpose. And then I could take that, because it's AMP resistance, and I can move that by P22 transduction into a recipient that's no AMBER suppressor, so it can't transpose, and now it has to go back into X by homologous recombination. Right? So, another version then is one where all of this is cut out to make it smaller. I think it's about 13 kb. So much smaller, but this is deleted. And so we use lac, and it was replaced with kanamycin resistance. This is what we're using in the lab. There's no transposase, but if you use a donor that nearby has an A+ gene, this can be transduced into recipient where you have the smaller transposon. This is called MUD-J. Important where the J came from. And the nearby A, because this is an unusual protein. Most proteins work in trans, so they can be complemented. So this only works in cis. It can't complement. So it has to be nearby, and it can recognize the ends and allow this to transpose, and then this just gets degraded. Okay. So now we get an insertion in the genome, right? There's no more transposase, so it can't transpose anyway. So we use this a lot, okay? And that's going to be useful for the paper on TPOS. Okay. So that brings the class, I hope, up to speed with the lab class already. Alright, so that's one transposon. That's these bud. So these just stands for defective. So if U is defective because it's missing the tail genes, so now we have to use something like P22 or another way to move it from one strain into another. Okay, that's teapot. So we mentioned TN10. TN10 has tetracycline resistance. Write it this way. And Nancy Kleckner made a derivative of this. So it's a composite. One of these has transposase. But Nancy Kleckner deleted all this and made a smaller version called TN10 defective TET. This is 10 kb. The smaller version is only 3 kb. But there's no transposase. So that means when we go into a recipient, which we're doing in the lab, we have a plasmid from Nancy Kleckner that has the transposase gene. Unlike you, this can work in trans. So you put on the plasmid, bring in that small transposon, and that will complement transpose X, and then we can move that into a new strain that has no transposase plasmid, so that DNA with the TN10 TET can only get in by recombination, right? Can't transpose. All right, so she did a couple versions where she replaced the inside, and the transposase just needs to recognize the ends, right? This is 10 to 20 base pairs at the ends is all it needs to recognize. So there's a TET version, there's a chloramphenicol resistant version, and there's a kanamycin resistant version. So she made several versions, TN10 TET, TN10 chlor, TN10 cam, so you can use different drugs. You have multiple things in the same strain. All right. So we like to use these two a lot. Not this one because this mud J is kanamycin. So we do a lot of things where we combine muds with these guys. There's only so many drugs you can work with. All right. So let's get back to TN10 TET. So the tetracycline resistance is the TET-A. And TET-A is an efflux pump in the membrane. So any tetracycline that gets in goes right out. And Stanley mentioned, well, having this in the membrane, the cells don't like it so much. And it makes the cell sensitive to fusaric acid. So there's what are called lipophilic compounds that get in the membrane that can kill E. coli. So if you have no TET, let's give you, I'm just going to make up a number. 100 units of fusaric acid kills. Okay? But if you have TET-A in the membrane, it might be 10 units kills. So you can have a condition where you can kill strains with the TET-A gene expressed, but not the wild type. Okay, so that allows you to select against that tetracycline resistance gene. We'll talk more about that later in the meeting. Okay. So, there's also a TET-R gene, which is the repressor, and the TET-R shuts off expression by binding to a region between the two promoters and shuts it off, unless you have tetracycline, and it comes off. And now you make the pump to pump out the tetracycline. So these promoters are turned on by the addition of tetracycline. Yeah? So this turned out to be pretty useful. As I mentioned, there was in the lab, there's a student in John Ross' lab. So every transcript, in the end of the transcript, there's a terminator. Okay. We actually talked about polarity, right? So the ribosome has since, when there's no translation behind it, you get these secondary structures. In a classic, there's two ways of terminating. There's 3' UTRs that have some C-rich regions that are recognized by a protein called Rho. So Rho will bind in the 3' UTR and knock off RNA polymerase. Okay, so that's one mechanism to stop transcribing a gene. Another mechanism is called Rho-dependent. And these are stem loop structures followed by U's. So this takes advantage of this natural evolution of RNA polymerase sensing upstream stem loops. Okay? So this is a natural terminator. So those are the two classic terminators. So what an undergraduate in John Roth's lab did was simply delete these terminators. And there's a strain that has the TET-A deleted. There's a strain that has the TET-R terminator deleted. And there's a strain with both. So there's TET-A, TET-R, and then both. So you can have transcripts that continue to read. Now if that transposon lands in front of some regulatory gene, and we add tetracycline, then we transcribe out from the terminator and now into this regulatory gene. Okay, so we can have a TET-dependent turn-on of some gene that normally may not be turned on. Okay? So this is called TFOP. All right, and this is what you guys have to be able to do to graduate. Okay. So John Roth came up with a song. In the States, or actually it's from England, there's a children's song called, it has to do with a teapot, right? And it's, I'm a little teapot. And then you, what is it? So when I'm, what is it?
Short?
Well, I'm just, what's the part, the end part? When I whistle? Anyway. So he wrote a song based on that, and that's the I'm a little teapot. Teapot. I'm a little teapot, I've got clout. Two promoters both point out. Tetracycline, hear me shout. I turn genes on all about. That's what you guys have to do together. We're going to film it so we can compete with all the other classes that are on YouTube. Learn the teapot song.
Can I just add to that? The biggest group we have so far is a class like this from Santiago, Chile. So, you know, it's Mexico versus Chile. Think of it as a soccer tournament.
That's right, it's a football match. With genetics. Okay.
So I have the letter, the lyrics here. You just need to cut it, or you guys can pass it out and get a picture in your phones. I think that's going to be easier then. So I'm going to pass it, and then it's divided in four, so it's only one. And my little teapot.
Okay, are we ready?
See?
Are we ready? So this is what we study in my lab, is how they build their flagella. Okay? They use flagella to swim. And the flagellum gene is called glycine. Okay? And there's like 20,000 subunits. That's a lot of protein. Each one of these is 1% of the cell protein. So that's a lot of protein. Okay, it's a lot of energy. So the cells have figured out they don't want to express all this protein until they can put it outside the cell. And so there's a regulatory mechanism that we work on in my lab that couples gene regulation to assembly. So there's, oh, and also I should tell you, the cell also has a nose. And there's both ends. Okay. So the cell is it's only 1 micron, right? So it cannot sense a concentration gradient across its own body. So the nose, every 2 or 3 seconds as it's swimming goes, smells good. Tacos are that way. But if it's moving along and it says, nope, tacos are that way. So it can switch. These are rotating counterclockwise, which is left-handed. And it can switch when the nose says, uh-oh, it's going to send a protein to the motors and it's going to go clockwise. So it's going to switch from going left-handed to right-handed. So when it's left-handed, it's pushing the bacteria. But if it switches to right-handed, it's going to pull it. And it's going to change direction. So when it goes clockwise, these flagella come apart, and he changes direction, and now he's going to go off and go back, and say, okay, tacos, this way, right? Or if it's still the wrong way, he'll do it again until he gets it right. Okay? So we call that a bias random. All right, enough about that. But the point is, there are 60 genes to build the flagella in the nose. That's a lot of genes, right? That's more than 1% of the cell. But fortunately, the regulation is a little simpler. So there's those 60 genes, but there's only 3 promoter classes. This is what we've studied in my lab. There's the class 1, which encodes an operon. Okay. And there's a lot of regulators, most of which we discovered using T-pops, which I'll tell you about. This forms a complex of 4 Ds and 2 Cs, and this goes to class 2 promoter. So class 2, like LAC, has a -10 and an upstream activating site. So sigma can recognize this. But this is recognized by EC, just like cyclic AMP, CAP were recognized in front of the LAC operon. And now this can bind to the alpha subunit of RNA polymerase. So you need this to bring RNA polymerase, which is alpha 2, beta prime, sigma. This is sigma 7. Okay, so we're going to turn on the class 2 genes. And the class 2 genes, sorry, they already said 2. It's okay. They make the motor. Okay, so if we look at the building of the flagellum, this is inside, there's a cell wall, this is outside. There's a ring of proteins that get put in there. It's a donut hole. It's like 34 subunits. It's a donut hole. It's going to put in a rotor. Okay, the rotor of the motor. And inside its membrane, because everything has to go outside the cell, they build what's called a type 3 secretion system. And so it's a member of a family of systems that secrete proteins from the cytoplasm outside. There's an injectosome structure that we talked about in the lab that also uses a similar secretion system. So now we got to get here to here, and they start building what's like a drive shaft, but they hit the cell wall. So they put on top of this a cap that has muramidase, and it chews a hole. Now we got a hole, great. Now we can chew a hole through the cell wall, and we keep polymerizing this drive shaft to the tip. We call this drive shaft the rod. And now it just sits and waits. So we made it to the outside, but we hit the wall. So there's another couple of proteins. The main secretion system in bacteria is called SEC. And there's proteins that come out using this, and they form these rings, and they punch a hole. Now we have a hole in the outer membrane. They knock off that cap, it's off, and they put on a new cap with a structure. So if I show you these structures, so I'm going to just draw it out here. Here's a structure. This is called the hook basal body. So that's the motor. So this hook is a flexible structure. It's like a universal joint. So this flagellum could be on this side of the bacteria, or it can be on this side, because of this flexible U-joint. It can be doing this, or doing that. Okay. All right. All right, that's good. So where are my genes? Out here we are. Okay. So we have the motor, which is this hook basal body. But in addition to hook basal body genes, there is a gene A which encodes an alternative sigma factor called sigma 28. And that brings RNA polymerase. So sigma 28, alpha, beta, beta prime, to the class 3 promoters. And that's filament, lysine, and the chemosensory. Okay? So all these things that are needed after the motor is built, right? The propeller. Okay? So, okay. So, initially, when I started working on this, I used these mud fusions. So for a class 3 expression, I had the Fly-C gene with its promoter that's dependent on this alternative sigma factor. But I fused a mud J, it's a lac. So when this gets built, these are lac plus, right? And when I first started my lab, what I found is if I broke this motor, if I had no motor, they had the wild type hook basal body, Fly-C lac, they're lac plus. But if I broke the motor, they're lac minus. So there was some mechanism that said, we're not going to make the propeller if there's no motor, right? And so how are we going to use genetics to figure that out? We have a condition here. What is that? What kind of a condition is that? If you're a geneticist, what do you see? What do you see? What can you do with that? This is what we call a positive selection.
No.
This is what geneticists dream about. This is what wakes me up at 2 o'clock in the morning. Oh, yes! Or sometimes, I remember once I was on an airplane, I go, oh, yes! Right? Positive selection.
They put them in handcuffs.
So we can select for LAC+. We can select for growth on lactose in strains that don't have this replacement. And we can do that following transposon mutagenesis. Right? So we can do a TN10D10, for example, with TET resistance, and then screen for LAC+. So we have a negative regulatory gene that is somehow turning off this. But now, if we get a TN insertion in it, we kill that negative regulation and we're LAC+. So that gene turned out to be one of the flagellar genes, of course. And so in addition to sigma 28, there is a gene called FLAG-M. Okay, and that was the negative regulatory. So a FLAG-M, TN, HBB minus, YC-LAG, is now LAC+. So that gives us something to work on. And we didn't figure this out, our competitor figured this out. A guy named Kazuhiro Kutsakaki. Don't mess with Kutsakaki. Alright, so. They're building the structure. They're building the hook base body. But they also have the sigma factor inside, being the with the hook base body genes, but FLAG-M. And FLAG-M is an anti-sigma factor. So it binds sigma factor. It'll even strip it off of RNA polymerase. You're mine. Okay? And so that keeps class 3 genes off because you need that sigma factor to turn on these class 3. So now let's build the structure. And now we have hook basal body complete. Okay. And when it reaches 55 nanometers, that's its terminal length. We don't have to go into details unless you want to. There's a signal to the secretion apparatus. It says stop, stop, stop with the hooks. We're done. Start with the filament. Yeah? But we need sigma to turn on the filament gene. But FLAG-M, so we transition from early, what we call early substrate specificity to what we call late, like Fly-C is a late substrate. Late. So there's a change in the apparatus that switches specificity from one set of substrates to another. To my knowledge, it's the only secretion system in biology that actually transitions from one set of substrates to another. M is a late substrate. So now when the switch occurs, E becomes the substrate, M is out, and now sigma 28 is free. And now turn on class 3, and now you make the filament, but only when there's a motor that's built. Okay. So that was my research career. All right. So you need that background. So we're interested in looking for new regulators using T-pump. So let's do this experiment. So we have a string, that's YC-LAC, YC-LAC-J. so the promoter of that sigma 28 dependent gene is now expressing the lac operon, right? As long as the hook basal body is good. And into that strain, we do a T-pop insertion. And we let that go into the chromosome, and if there's some regulatory gene, T-pop, with TET plus TET, we now can turn on that regulatory gene. So, we do a T-pop hop, we select TET, select. And then we look for TET lac plus or TET lac minus, no TET lac minus, no TET lac plus. So this would be a condition where we found a negative regulator. So when we turn it on, this is shut off. Or we land it, perhaps there's a positive regulator. Where now we've inserted the T-pop. So that promoter can't get through because remember transposons are polar. So he is no longer positively regulating, so they're lac minus. But now we add TET, and now we can turn it on. What's the other way? Yeah, turn it on, yes. Okay? So that allows us to identify regulatory genes. And these regulatory genes may be turned on under conditions we don't know. Okay? But it gives us things to figure out. So what did we get? And that paper is just one example of some of the things that we get. But I will list what we got. Okay, let's see. All these regulators. What did we get? Where did we get the T-pops? In front of the FLHTC genes that knocked out the promoter. That's a case of TET turning on the system. That makes sense? What else did we get? Same thing. Fly A. Sigma 28. If you have an insertion, it's now off, but we can have TET to turn it on. Flag M. So this, if you overexpress it, even if there's sigma 28, if you have now excess, it'll be a case where it'll turn off. Because it's inhibiting that signal. And then what else? Oh boy. Oh boy. Oh boy. CAP, CRP, RFLM, RFLP, LID, LRHA, SLIA, HILD, LD, and this RTSB, and MZ, FI. Did I miss anybody? Oh, CSRA. Holy cow. All right. So now we got 2 decades worth of work to figure out how are all of these guys regulated for gelatin expression. Only going to talk about a couple. Right? So actually, within the class 2 is something we discovered called RFLM. So when these turn on the hook basal body genes, you make a set of hook basal bodies, but they do turn on an auto repressor. So you just have a burst and then off. I think in the paper we talk about, let's see, RTSB. That's one. And we talked about this. Oh yeah, this is the name in the book. We renamed it. It's called ECNR. And on the chromosome, there is a, what do you call it, toxin-antitoxin system. And right next to it was a regulatory gene. It, you know, your bioinformatician said this is a repressor. I don't know if it's a lysar repressor or something. And since it was next to these genes, they just assumed, oh, it must be regulating these genes. It doesn't. It doesn't. So that's why it had a bad nomenclature. So don't always believe what you read. It turned out that that was our regulator of flagellum. And I'll just talk about two. Well, we can talk about Hildy, you might be interested. FIMZ. FIMZ is an activator of fimbrial gene expression. That's right, I can't spell fimbrial, it's got a weird name. Okay, so when you're turning on fimbriae, if you activate that, it turns out he represses FLHDs. So he binds that promoter. So if you want to swim, swim. But if you want to stick, so fimbriae are used to stick. You don't want to swim away. So that activator for fimbriae shuts off flagellum. This is actually another fimbriae regulator, just like FIMZ. We get that. Okay, what about, what did I say I'd do? Well, let's do LRHA. So LRHA was discovered by Tom Solhavi, not knowing what it is. Lysar homology. So some kind of a regulatory gene that looks like this lysar repressor. Lysar repressor, LRHA. That actually shuts off again. And for decades, it was unknown what that is, right? Until recently, it was shown that this responds to low pH. So salmonella, when it's engulfed into a eukaryotic cell, and it comes into these salmonella vacuoles, this is a low pH. So LRHA is turned on what's called the SPY2 conditions, but nobody knew why. Turns out that flagella at low pH, these dissolve. And that may be a mechanism that when you get inside a cell, this is the very abundant antigen. The immune system goes, whoa, we got bacteria, let's go kill. But if you can dissolve your flagella, then they can't see you anymore. So it's a way of saying, okay, let's shut off and not make them if we're in acidic conditions where they're going to fall apart anyway. And then what about this guy, RTSB? So RCSB, sorry. I don't want to do that one, I'll do RCSB. It's not even on there. All right, here's another one. So if you have membrane damage, that turns on this RCS signaling pathway to cell to cell. Hey, we got damage, let's go turn things off like FLHDC. We can't build a flagella, a hook beta body, in a damaged membrane, so that turns off FLHDC. Right? So you got to find all these regulators and make all these discoveries. All right, Hill D. Why Hill D? That's a good question. Okay, so far so good. Several that were in the paper. You can ask specific questions if you want. see, this allows you to find regulators, even if you don't know the condition that they're normally turned on. That's why the teapot is so great. And you gotta go figure it out. That's the hard part. Okay. So those of you probably know that Hill D is the main activator for type SPY1 injectosome production. So salmonella has these injectosomes, SPY1, that allow it to get engulfed into cells, these injectosomes. There's a lot of regulatory conditions to turn on or off Hill D. Hill D turns on Hill A. Hill A turns on injectosome. And two key regulators, NF and SIG-A, which turn on effectors. So we landed a teapot in front of Hill D. And we saw that no tech, it shut things down with, let's see if I got this right, with no tech. No tech. Yeah, here we go. Sorry, so no tech with black mice. So we disrupted its normal promoter, and now we add tetracycline, and then we can turn on Hill D, but we also turn on flagellum. Huh? Okay, so we're gonna end this just to show you how these all work. All right. You take an overnight culture, and here the flagellar system is off. Law is off. Once you hit saturation, everything gets turned down. But if we dilute these into fresh media, what happens is there's a promoter that turns on FLHDC. Right? Okay. Fresh media. Boom. We call this P5. And now we make a hook basal body. And I mentioned that there's the fly A, sigma 28, and M. There's also proteins that have to get put on before the filament can polymerize. And these are hook filament junction, and then the filament cap. Those have to get put on. So we have the hook, filament junction, and the cap. So in type 3 secretion, we have early substrates and late substrates. So one thing that differentiates early substrates, these guys, from late, like the filament, and these, late, is they require a secretion chaperone. That is a helper protein that can bring them to the apparatus to be secreted. That's all effector proteins and injectosomes, they need a secretion chaperone as well. And the hook filament junction proteins have a chaperone called N, the cap has a chaperone T, and by having these ready, they're protected from proteolysis. But remember that filament, that's 20,000 subunits. That's a lot of protein. Cap, there's only 5. This, there's only 11 of these two. Not very many. And these have to get out before the filament. So by having them already pre-made, it can't go out until the switch happens. Switch. Now they can go out. Out. They can assemble, or the M is out. And when they're out, they release their chaperones. So these chaperones have day jobs and night jobs. Okay? The day job is to protect their substrates until the switch occurs, and then guide them to be secreted. The night jobs is regulation. We talk about sigma 28. Sigma 28 actually is the chaperone. Flagellar. And when Flagellar goes out, now we can turn on class 3, like C. N actually gives you more M. It's a translational regulator. Actually start producing more M, because eventually you're going to want to shut it off. T is actually targets DC proteolysis. So that way, again, you shut off hook basal body production while you're building control. Okay. And this was one of the, if you have a T-pop in front of T, it shuts everything off because you're always proteolyzing. Yes?
Is there a way to regulate the number of subunits in the agilum?
There is a way, absolutely. And the way is to make more DC. So if you make more DC, you can, instead of 4 per cell, normally there's 4 per cell on average, and we get it up to 15 by actually, and sometimes we can just knock out some of these negative regulators to increase it, or we can just overexpress DC. So that seems to be where the number control occurs. Anything else? Okay. Am I done with T-pop? What else? I think I'm done. Okay, so that's. I just want to show you how cool this is. Right? So you find genes. You know, you can do a microarray, you can do an RNA-seq, but if you don't know the conditions, what are you going to do? We just have to add tech. We find them, and then we got to figure them out. Okay, so that's paper number 1.
Well,
Are we going to stop?
Yeah,
We're
yeah,
done. Oh,
it's time.
all right. No more
We have a
Q&A.
time limit today, so we need to get
All right.
out.
I mean, we started a little late. You
Oh.
want to mention the
Yeah,
second paper?
you just wanted
Oh,
to
I
talk
forgot.
about one of those.
Oh, wait, I forgot. Last thing I gotta tell you. So the phi A sigma is in an operon with Z. Z actually activates D, and D actually turns on another promoter. Actually, this is P1. So as the cells are growing, time, you have the P1 turning on FLHDC. And later in the growth phase, you have P5 that's dependent on hill D. I have no idea why. No idea.
Might be related with the fact that you remember these studies that they start the spike 1 inside the cell.
Yeah.
That might be depending on, and then they express the flagella. I remember that from her studies.
So that would be a good.
That
Yeah,
would be
I'll
interesting
look
to
into that.
measure
Yeah.
if it is working in
Yeah.
that from our especially
Correct.
inside the cell.
Yes. Okay. Thanks. What you do that? Okay.
And the second paper we might move to tomorrow.
Oh, okay.
All right.
Because all integrates the challenge stage paper into my talk.
So it's a job for questions. Well, we didn't go through the whole paper. I guess you are relieved all.
Well, you get the main point, right?
Yeah, that's the important thing. But I wonder if there's any questions besides Professor Everardo. Other questions about the paper?
Well, this methodology can works with if you are looking for a master regulator, not like in the end of a test with regulation. FLHDC.
Yeah. So I hit FLHDC with T-pop or anything that regulates that. Also sigma 28. If I get a T-pop in front of that, like that. So can get anything that's going to regulate my final output. I mean, if I put the mud in a class 2 promoter, I would not have gotten sigma 28 or flag M. I would only gotten DC.
Which are the limitations of this method?
Which are what?
The limitation.
What are the limitations of this method? What are the limitations? What are the limitations? Boy, I've never thought of that. Can you think of the limitations?
Well, I think that if you were in a different organism.
Oh yeah, of course.
Yeah.
If you're an organism that you can't, you'd have to find your own induced promoter with a transposon. And we actually, the bulk of their paper, we had a promoter that was inducible, but it was not inducible in Procolaria. It was inducible in E. coli. So these things don't necessarily work. You have to find the promoter, inducible promoter for your bacteria. For sure.
Any other questions? No? Okay. Well, thank you very much for today. So, some, how do you say avisos? Warnings. Well, so the people who's doing the lab, we're going to take a break from, I guess, 20 minutes, a little bit, so you guys can have something to eat. And then the rest, the people that let us in is asking us to leave through gate 5, la puerta 5, por ahí salimos, los que van a salir ahorita a la mitad. Los demás nos quedamos para terminar de una vez el laboratorio y no tener que salir y entrar otra vez, entonces de una vez avanzamos eso y terminamos más temprano hoy. Y por eso vamos a dar 20, 25 minutos y ya nos vemos en el laboratorio 4 para continuar. No sé si quieran, sí, la idea es con la cancioncita esta, bueno, que ya la fotografiaron, ya tienen por ahí, hacia el final del curso, pues bueno, van a tratar de dirigir, les voy a mandar unos videos. Vamos a tener que demostrar que los mexicanos son más entonados.
We have the lyrics, but what about the music? We have the lyrics, but we don't have the music.
You want to practice right now? Just the lyrics.
Oh, yeah, yeah.
Singing.
A little
Okay.
sing along.
Okay. Does everybody has this? Have their phones? Okay. This way.
Just the singing.
Just the singing. You want to do the dancing? We can help you.
No, I don't know that. Only if someone comes up and dances.
Who wants to come and dance with the professor? Come on. Come on, Abby.
Not yet.
See? Ok, dos valientes. Tenemos dos valientes por aquí. Y muchos teléfonos listos. Ya lo sigues a él. Uno más.
Uno más. Orlando. Orlando también, Orlando.
Fall off the stage.
Okay.
Ready? I'm a little T-bone. I got clouds. Two promoters both point out. Be tetracycline, hear me shout. I turn genes on all about.
So tomorrow we'll practice some more. Mañana depende de lo que pase con los compañeros de horarios y la escuela. Entonces igual hoy fue todo muy aventurado en la mañana, muy rápido. Afortunadamente dejaron pasar a los compañeros del video también, nos dejaron pasar a nosotros. Entonces creo que si llegáramos a seguir con la escuela cerrada, pues haremos lo mismo, entrar por la puerta 5, nos vemos. Tal vez podríamos empezar un poquito más temprano, nos podríamos ver a las 9 para entrar y poder tener todo listo aquí. Y no sé si hay alguna pregunta. ¿Any question? ¿No? Bueno, gracias a todos.